William Pauli. Sorry if it's Duke. You are an assistant professor with the School of Plant Sciences at the University of Arizona, interested in all aspects of agriculture and agrosystems, and focusing on understanding the genetic basis of stress adaptative traits in crop plants through various mechanisms. So we're going to be a little less broad on the topic of tipping points and focusing on one topic, and you're going to tell us about leveraging high-resolution Phenomics data to address crop performance uncertainties.
Perfect. Thank you so much. Well, good morning and good evening, everybody. Thank you for your time. Again, my name is Duke Pauli. I'm professor in the School of Plant Sciences here at the University of Arizona. My lab is specifically focused on understanding the genetic basis of heat and drought stress tolerance in crop plants. And basically how we can use, crop, genetics and whatnot to basically prepare for these, the tipping points that have been discussed so far. Before I begin, I would just like to thank the organizers for the invitation to present our research today. I'd also like to take the opportunity to thank all of our team members. This project has been quite huge. It's spanned about 6 years, and they've done a ton of work, and I'm just the fortunate one that gets to present it. So today I'll be talking about our research using the field Scanalyzer. It's this giant robot in the background that you see here. This is the world's largest agricultural robot. It covers about 2 acres of land, and that glowing red box basically houses a whole bunch of sensors that I'll be talking about. And we're using this tool to basically try to unravel the dynamics between the genetics of the plants and the environment itself. And so again, my presentation is a little bit more focused in terms of using new technologies in conjunction with machine learning, artificial intelligence to understand how plants respond to the environment and using these types of data to predict plant performance. Additionally, Arizona is one of the best places to do this. It's consistently hot and dry. Last year, from the month of April through October, we got 2.5 millimeters of rain. It's always sunny, it's always pretty hot. And so I always tell people that, we have the climate of tomorrow today. And the reason I bring that up is, next slide, please, is that we know that agricultural production is really threatened by variable amounts of water, variable climates, and there's more competition for the freshwater resources that we do have. And so this poses this really great question of how are we going to continue to increase our agricultural productivity when we have less available resources. Further compounding these problems is that, globally it's predicted that more of the earth will become arid or semi-arid in the next 50 years. A significant proportion of our arable cropland will be impacted by this. And so basically, we're facing this major challenge of, again, how do we feed a growing population with less resources? And so this is kind of in context of the, series of tipping points is that, our ecosystem is reaching this point where it's going to shift, where more of our land will be dry. We won't have the water. So how do we prepare for that? And that's kind of where our research fits in, is that, crop breeding takes decades to release, improved varieties that can cope with these different stresses. And so that's kind of what our research is focused on, is beginning to prepare for that now, because once it happens, once we cross that tipping point, it'll take us such a long time to catch up that we don't want to be caught flat-footed and not have, plants that are able to withstand this new environment that we'll be growing in. Oh, next slide, please. And so central to this whole research question is the idea of connecting genotype, the organism's genomic makeup, to the phenotype, so how it actually performs, or its physical performance. The drought phenotype is rather complex in that it's a whole plant response. And additionally, it's a response that relies on the environment in which it's expressed. There really is no single gene solution to drought tolerance. I know some of the major seed companies have marketed that, but realistically, we haven't seen that come to fruition. So because of that, improvement must be at the whole plant level. And so it's really understanding how the biology and the genetics of stress adaptive traits manifest into superior plant performance. And so our work really seeks to integrate physiology and genetics into understanding how these two components interact to give rise to a phenotypic landscape, which is on the right-hand side. And so just to walk through the figure, so genetic information can really be considered binary, bit information. Instead of zeros and ones, it's A's, T's, C's, and G's. And because of this, it's often thought that, genetic data resides in a two-dimensional space. But as this, information is translated through the organism, it interacts not only with the internal environment, if we think about metabolomics, proteomics, RNA-seq work, but also interacts with the external environment. And so it gives rise to the shifting landscape where one gene can have multiple phenotypes depending on the environment in which it's growing. And so if we think about this in the context of drought research, there are different dynamics, like the soil in Arizona is much different than that in the Midwest. And so a gene that might confer drought resiliency here may not work in another environment. And so really understanding and teasing apart how that works, that dynamic between the organism and the environment, is so critical. And again, that's kind of what we're focusing on so that we can begin preparing and developing the plants that are going to be needed in the future now. Next slide. So the Gantry is this large, basic science research tool that you see in the background behind me. It is the world's largest agricultural robot. It's like 30 tons, it's massive. And because it's so large, it can carry this box of sensors. So we have things like a 3D line scanner, we have a thermal camera, we have a camera called a chlorophyll fluorescence 2 camera, which actually quantifies the photo efficiency of crop plants. And we have hyperspectral sensors. And so basically the issue that we're facing is that this instrument generates, 10 terabytes of data per day. Our typical crop season is 90 days to 120. So we obviously don't run it at that rate every day. Our typical performance is about 1.5 terabytes of data generated per day. And so to give you an idea of the data products that we're looking at, on the left-hand side, we have like your standard red, green, blue photo of a crop plant. The next image is thermal. So this tells us how warm the plant is, and this is a proxy for stomatal conductance, how the plants are transpiring and cooling themselves, and basically relating to photosynthetic capacity. Next, we have chlorophyll fluorescence. Again, this is a sensor that measures how well plants can convert energy, sunlight energy, into biomass. And specifically, it's capturing how electrons are moved through the electron transport chain during photosynthesis. One of my favorite sensors is the 3D line scanner. It generates these very high resolution images of crop plants so we can get 3D structures and basically have images of plants that can last, for decades later, so we can come back and analyze them. And then finally, the one instrument that gets people the most excited is hyperspectral. So basically, this is a camera that takes an image at every wavelength from 350 nanometers all the way up to 2,500 nanometers. There's a lot of promise in what this data can give us, but we're still trying to unravel like what that is. So these are the sensors that are on board, and these are kind of what we're using to study how crops respond to the environment. Next slide, please. So one challenge that we've really had to overcome is the massive amount of data. So in a season, we can generate 300 to 400 up to 500 terabytes. And this was problematic because, after 2 crop seasons, we're at a petabyte of storage, or data that we have to store. And to this point, this project, we've generated enough that we've moved, I think, 1.5 petabytes to magnetic tape. So we had to come up with a way to process this, and we have a pipeline called PhytoOracle, which is an open source, scalable, modular workflow. And basically what happens is the Gantry completes a scan of the field. The data is packaged at a server on site here at the research station, and then moved down to Tucson, where it's uploaded into the data store at Cybers. From there, a master node, basically the computer that's in control of everything, distributes the data and the commands to worker nodes that then sit there, compute on the data set that they're given, and then send the results back to the manager node where everything is compiled and then reported back to us. And the reason I bring this up is because this was a significant challenge. Using a standard, 4-core lab computer, processing just one full day of data would take us about 11 years. Obviously, that was not going to be a tractable solution. So with PhytoOracle, we can basically trim this down to a little over a day. I'm not the data expert, so probably I can answer some of your questions, but not all of them. But what we're interested in is the data products that come from this. Next slide, please. So the first application of machine learning, artificial intelligence that we had to face was, believe it or not, the Gantry, despite how high resolution it is, can have issues with taking images. It's windy, that box can blow around like basically a pendulum. And so you see on the far left-hand side that we get like the staggering effect in our images. Conventional software for doing orthomosaicing, such as like Pix4D or Agisoft or something, is not able to handle this because there's not enough overlap in the images. So we reached out to computer scientists and basically developed our own machine learning algorithm called Megastitch, which is able to process and align these photos at a rather rapid rate. We're actually able to outperform commercial software. And so you can see on the right-hand side, this is what a finished field looks like in terms of having all of our 15,000 images perfectly lined up. Next slide, please. Why this was such a challenge for us is that we're outside operating in an environment and we have environmental constraints. So we have issues with, clouds rolling over, which is what you see in the left-hand side, where half the field's illuminated and another portion is in the shadow. Additionally, on the right-hand side, the Gantry in and of itself is kind of a constraint because it casts a shadow on the very things that it's imaging. And you can see that there's this constant shadow in the image. And so our algorithms must be able to handle the inconsistent illumination conditions in order to stitch together all these images. So that was kind of like one of the first challenges we faced. And this was critical for us because of the step that came next. So next slide, please. So what we're really interested in our acre and a half of land is, I want to know what's going on with each and every plant. Typically field-based research for crop genetics has been based at the plot level. We just average across all the plants in one plot. However, that's not always illuminating, and really, understanding more of the dynamics of the crop growth was really the target. So we developed a faster RCNN convolutional neural network to basically identify these plants, locate them in the field, tag them, and then for each successive day that we're collecting data or scanning the field, compile the data across the entire season. So in this case, we just selected 5 days for the purpose of the slide. And so after the day, we can use another machine learning approach, agglomerative clustering, to basically connect all the data points across the field. And why this is so critical is because now, for all 30,000 plants that are in the field, we can track one individually. We can look at how it grows across time. The team's made some very cool movies of individual plants growing and responding to the environment. But we can capture the dynamics of crop response, i.e. we know crops grow like this, in a sigmoidal fashion, but in terms of how fast, they respond to environmental fluxes. So in this case, we were studying the impacts of water deficit on lettuce growth. And that's really what we're trying to target, is understanding the response of the crop to the environment. And that's why being able to track all these individual plants over the entire season was so critical for the work that we're doing. Next slide, please. Another thing that we know it's going to be prevalent in future agricultural scenarios is plant diseases. They're becoming more prevalent. Because of our monoculture system, we've basically created, artificial selection environments to create more, robust pathogens that we have to deal with. And so these are just kind of an inevitable aspect of, agricultural productions. So we really want to focus on having tools to detect and aid in management decisions. Typically, we speak of, management as reactive. We see the problem and we're struggling to catch up with it. And so having tools like this allow us to shift to a proactive mode where we can be more prescriptive with how we manage our crop. And so the specific disease that we face is called charcoal dry rot. It's caused by a genus of soil fungus called Macrophemonia. Macrophemonia, there's 200 different species. It impacts about 100 different crop plants. It's prevalent in dry, arid soils. So basically the soils that will become more prevalent in the future, as climate change continues. So this is kind of one of the things we're working on. Again, developing an artificial network, so neural network, to basically detect and quantify the amount of disease. So on the left-hand image at the bottom, you can see a plant that's struggling. You can see the dry, desiccated leaves and some red marks. The red marking is basically a human going in, identifying the disease lesion areas. The middle image is what the computer identifies as dry rot. So this is its best prediction of identification of dry rot in the image. And then finally, on the far right-hand side is when we overlay the two images, intersection of union is the metric that we're interested in. We can compute an average and say, how accurate is our algorithm? At this point, we're operating at about 95% accuracy. And why we're excited about this is, yes, not everybody has a gantry, but UAV technology is becoming quite prevalent in the agricultural space. And so right now we're working with NVIDIA, and specifically some of the small GPUs they have to put them on the drone so the drone can fly, capture images, and then in real time process, identify, quantify the disease presence so that when it lands, it can basically report out to a farmer or crop consultant, this is where the disease is at, this is how bad it is. So that, farmers can make more proactive decisions. That's probably one of the biggest problems with the adoption of technology, precision ag technology in the workspace, is that farmers are busy. They don't have time to upload data, process, analyze. They want a tool that can just give them answers on the fly. And so we're hoping to develop one of those. Next slide, please.
You have less than 1 minute.
Okay. So this is some really fantastic work we're doing with two very great people, Dr. Bedrick Beans at Purdue University and James Snobble at UNL. What we're interested to do is growing plants in silico. So we're using our 3D point cloud data to understand plant growth over time. So you can see in the photo, we've basically grown synthetic genotypes of plants that have different leaf angles, morphology, and we're actually using that to understand and simulate organismal development in relation to the environment. So basically modeling plant growth in real time. One reason that we're interested in this is to understand how light interacts with the plant canopy. Computationally, we can do like a ray tracing experiment to understand how light is intercepted at lower levels beyond just the top of the canopy, and how the light use efficiency relates to biomass accumulation and production and overall yield. The next slide, please. And finally, the last thing that we're working on is how do we leverage all these data to make predictions to help us understand how changes in the environment impact agricultural production. So on the left-hand side, you can see one of our 3D renderings of a early lettuce plant. And so we have, like I said, we've collected this data throughout the season. And so on the right-hand side is a graph of basically the true value of yield, so the blue square. The predicted is the green cross mark. And then we have the data points throughout the season. So we can see that plant growth looks, as we expect, exponential. But if you look at where the line goes, it doesn't really intersect with the final yield. So there's a nonlinear trend. So we've been using long short-term memory networks to basically predict crop performance. And so this prediction, so basically we've made this prediction 30 days out. So 30 days before we would harvest the crop. And our model is able to predict with about plus or minus 5% accuracy what the final yield is. And so anyway, these are kind of the types of tools and technology that we're using to understand the crop responses, to begin preparing for the future, and really start developing more stress-resilient crop plants. Next slide. And so I just wanted to conclude by thanking everybody for their time. I know my presentation is probably a little bit different than what we've seen thus far, but this is kind of how our group is starting to think about these tipping points and how we can best prepare for them. So thank you for your time.